Very much for that introduction, Nikki. So the advantage of being the last speaker, you don't have to go through the introduction and describing things because I think it has been described quite well. But I'll be presenting on the gut microbiome in African, which on behalf of the AWI-Gen Collaborative Center. Sorry. So you've probably heard earlier what a gut or what a microbiome is, so I'll not go over that. However, one thing that I can highlight out is there are different sites or different locations on our bodies. We have the lung, we have one in our skin, we have the gut, as well as we have the women reproductive systems. So one thing that I can talk about in terms of the digestive tract, which we now refer to as the gut microbiome, is it encodes more genes than the human genome by itself. So sometimes we refer to it as our second genome. So I'm sure you've heard more about some of the roles or the crucial roles. Hi, is that better? Sorry, it might be because I'm having. So you probably heard about the role of the microbiome in health. It's important in the immune system maturation, important in drug metabolism, as well as infection, fighting all these pathogenic infections. And we have a very complex, co-evolved relationship with the gut microbiome. We talk about how important it is and what affects the gut microbiome is what we eat, the lifestyle that we live, as well as the antibiotics that we're taking, and the diet, as well as the water and hygiene aspects. So the balance and the composition of the microbiome now can be affected by this balance of the good and bad bacteria, which then leads to disease statuses. So because of the disruption in the microbiome, which has previously been linked to several diseases such as inflammatory bowel diseases, as well as obesity, cardiovascular, and neurodegenerative diseases, there's a lot of interest in this microbiome space in relating to disease. And I'm sure most of you have heard of or watched this documentary on The Secrets of Your Gut, or be it You Are What You Eat, which is a UK twin study. But majority of what we know about the gut microbiome in relation to disease and health has come from the high-income countries. And this is because all this current microbial research largely reflects on these populations with these Western lifestyles and industrialized populations. And this limits our understanding of how does the gut microbiome in these low to middle income countries then interact with the microbiomes and under different environmental as well as nutritional conditions. And this has affects our ability to be able to develop equitable health solutions. So this is an example of a figure that shows how underrepresented African is in the microbiome studies. Okay, sorry. I don't know what's going on. Okay, great. So this is where the AWI-Gen study was able to make quite an enormous contribution to the microbiome field. And as described previously, the AWI-Gen study was set up to investigate the genomic and environmental factors that contribute to the cardiometabolic diseases. And Pehelo had already highlighted that it was collected in 2 waves of longitudinally. And the gut microbiome study was part of the second wave of this study, and we were able to subset these participants. So the study was conducted across 6 African sites, which is Nanoro in Burkina Faso, and then we have Navrongo in Ghana, and then Nairobi, we have these informal settlements, and then we have Agincourt over here in Mpumalanga. Then we have Dimamo in Limpopo, as well as Soweto in Johannesburg. So these sites, the reason as to why I'm highlighting them in this aspect, it just represent how they vary from rural to peri-urban to very urban communities with different diets, different environments, and health burdens. So we leveraged existing health demographic surveillance systems to be able to collect samples from these participants. And we had around 1,800 women enrolled into this study. And the reason as to why we had women is because we were leveraging or we were partnering with the menopause study. So 40 plus women were recruited into this study, and shotgun metagenomic sequencing was performed on the stool samples. And then we integrated this data with this deep phenotypic rich data set that we have currently, because now we have the demographic information, we have the health status of these participants, we have biomarkers from the blood and urine, and now we were able to integrate these with the taxonomic profiling and metagenomic assembly, as well as the functional pathway data. So one of the questions that we hoped to be able to address was what is considered as a normal or healthy gut in the African populations, and be able to understand how does the gut microbiome then differ across the African populations. So we were able to observe this high microbial diversity in the rural sites, which is the first two, Nanore and Nevrongo. And this supports the previous research that then suggests this industrialization that reduces the microbial diversity. And this gradient that we are seeing from the rural to the urban sites, and is not only very linear, because a site like Nairobi over here, which is considered urban, had this very mixed signal that reflects the rural to urban migrations. But most importantly, we saw that each site has a very distinct microbial signature as well. For example, we have this core bacteria, which was consistent across all the sites, but there are some of the species as shown in red, which are then found in more in the rural guts, which are the top side, and they're always lost in the urban population. So it shows how local diets and environmental exposures also have an impact on the gut microbiome. So we then looked closely at specific microbes to see which ones are the ones that are varied across the sites. So just to highlight, like Treponema, which is always lost with industrialization on my left. It's more abundant in the rural populations, as shown over here. And we can highlight also the difference, the inverse, which is Bifidobacterium, which was mostly found in the urban guts. And why we could see Treponema mostly in the rural population, because of less antibiotic usage in these rural sites. And these shifts then mirror these dietary transitions from high fiber traditional foods to these processed diets that we're seeing in the urban sites. So to be able to understand Treponema succinifacins as a marker of microbiome transitions, then we looked deeper into what are some of the factors that do influence its presence. And individuals with recent antibiotic use were significantly less likely to harbor the T succinifacins, which is this figure on the far right. And then the presence of the species was also linked to a lower waist to hip ratio, which is this figure here. And finally, we show that T succinifacins carries a wide range of these glycoside hydrolysis, which is the figure on the right side. So we also then hypothesized, probably because of the underrepresentation of African in these large-scale microbiome studies, then there'll probably be some microbes that have not been identified, or they're not in these databases. So we then constructed over 69,000 microbial genomes and de-replicated them over 2,600 high-quality mags from the shotgun reads, and compared these mags to the already existing database. And remarkably, we have over 1,000 genomes that were not found in the existing global reference database, and it underscores the microbial novelty in the African populations. So besides the bacterial genome, we have the viral genome, which we found over 40,000 novel viral genomes. And we also have some few new jumbo phages that were also identified in these populations. And this figure just shows that we are yet to be able to exhaust this discovery in these populations. Hence, the importance of being able to include these diverse African populations in this large-scale research for microbiome-related discovery if we are ever looking at precision medicine. So our research also aims to understand just how does the gut microbiome then relate to the disease status. And we focused on a subset of individuals with HIV data from South Africa and Kenya because we were powered because of the HIV prevalence, but also because of biological reasons, because the gut tissue serves as a reservoir for HIV, and it causes that damage to the epithelial barrier, hence leading to this translocation of the microbes across the epithelium. But antiretroviral therapy has been shown to be not sufficient enough to restore the gut remediation. So across Agincourt, Soweto, and Nairobi, people living with HIV had a significantly lower microbial diversity even when on antiretroviral therapy. And this just shows that some of those crucial microbes are being lost and plays an important role in the role of HIV in disease progression. But when you looked at now in terms of which taxa are differentially abundant, we find that over 130 taxas were associated with HIV status, and there was enrichment of some of the pro-inflammatory microbes like Fusobacterium in the people living with HIV, and Faecalibacterium, which is an unknown anti-inflammatory, was depleted in people living with HIV. But some taxa that have, which have not been associated with HIV previously, like Enterocluster as well as the Sosmobacter wilbionis, were also identified as very strong signatures of HIV. But these microbes, which I'm highlighting in black here, have yet to be described in the case of gut microbiome, and we're yet to find out what's their importance in HIV. So in summary, this work is published in Nature, and we are humbled that our woven mat from the corner plots made it to the Nature cover of the issue. And this so far is the largest gut microbiome study in Africa, and we uncover microbial novelty as well as the signatures of disease. But moving beyond the data that we found, we have this unique component about this project, which was returning of the findings to the participants. And it's important because we're able to engage with these communities and build these very long-term relationships as foundation for trust. So we were looking at it with a blank slate where it comes to returning of results to the participants, and we're talking about non-clinical data or non-clinical findings that we are communicating. So we had to be wary and try to explain to them in a very layman's language so that they can be able to understand as well. So this is slightly different from the way other populations do this type of studies. Because of the convenient sampling done in other populations, we were able to engage with the communities. So we were able to provide this personalized report. As you can see, this report is already translated in one of the local languages on one of the sites. This is in Kiswahili. So it's a 2-page report whereby we are able to tell them what's a microbiome, what are some of the things that affect the microbiome, what are some of the foods that are good for you, bad for the microbiome, and such like things. On the second page, we are able to now give them these individualized results. So this just shows an example of how our team delivered the results across the various settings, ranging from one-on-one discussions to group discussion, large group discussions, and focus group models. And each approach is very flexible to the population that we were working with. And we had an actual educational session explaining to them what's a gut microbiome and why is it important, are the bacteria good or bad, and such like things, and what can you do to improve your gut microbiome. So apart from sharing the findings with the participants, we also wanted to find out from them, did they even understand the type of results that we were presenting to them, or is there anything else that we can do better in terms of responding to these kind of questions. And some of our findings are just in terms of, it reveals the sense of empowerment among the participants. And majority of them shared that being able to learn about the microbiome was helpful in terms of connecting it with what they eat, the kind of lifestyle that they live, as well as their health. So one of the comments was like, I did not know the bacteria were good. Majority of them I know were bad. So you kind of can explain to them why some of them are good, and majority of them are good, and some of them cause diseases. So through this process, we've learned a lot, and there's importance of tailored engagement, and critically, there's need for clear budgeting from the start. Those are some of the lessons we learned. But community engagement is something that's feasible, and we should be able to incorporate it in our studies in the near future. And with this rich phenotype data, we're now able to look at other aspects of the gut microbiome in terms of health and disease. We're able to further look at the gut microbiome and the hormones, which is the menopause aspect. We're able to look at the neurodegenerative, the cognition aspect, gut microbiome, and the antibiotic resistance, as well as the host genetic aspects. So with that, I would like to acknowledge the AWI-Gen PI, Professor Michelle Ramsey, the study participants, the funding which made this work possible. And this work is a joint effort in collaboration with Rift University and Stanford University, and led by Professor Scott Hazelhurst and Amy Barth, University of Stanford. So we're excited about the future of directions of microbiome-related studies. Thank you.
It's a great exciting study. Question? I mean, we're back out of time, so
just quickly. So obviously, it seems like most people responded positively to the feedback. But do you have any idea if they actually implemented the feedback? Like, because obviously there's also certain costs associated with eating healthier, I assume. So do you know if they actually implemented the benefit?
My guess is as good as yours. We wouldn't know. However, we try to give them those small changes that you can make on your diet, not dramatic changes like, no, you should buy brown bread that is high in fiber. No, just change from white to brown. Dilute your juice or your soda with water. So those implementable changes that we know because of the socioeconomic status of these participants, we have to be wary of what we are suggesting to them as you should be wary about this, but you can change this around what you eat. Eat more high fiber diets and nuts and such like this. Yeah. And reduce on A, B, C, D.
okay, thanks.